Welcome to our Parasitology lecture. Today, we are going to explore a fascinating yet highly destructive stowaway that might just be the ultimate survivor, the giant human roundworm, scientifically known as Ascaris lumbricoides. Imagine hosting a dinner party, but an uninvited guest eats your food before you can even digest it. By the end of this session, you will understand exactly how this master thief operates and how we use mathematics to stop it. To begin our investigation, let us look at what this creature actually is. In the medical community, this parasite is formally known as Ascaris lumbricoides. It belongs to a massive group of unsegmented worms called nematodes. If you saw one, it might look like a common earthworm from your garden, characterized by a smooth, cylindrical, and tapered body. However, it is highly specialized to survive the harsh environment of the human gut. In fact, it is the largest nematode that parasitizes the human intestine, with adult females reaching up to 35 centimeters in length. Understanding its basic biology is our first step in learning how to combat the infections it causes worldwide. Now that we know what this parasite looks like, how does it actually get inside us? The disease caused by this worm is called ascariasis. It is primarily transmitted through what we call the fecal-oral route. This happens when microscopic eggs, which are invisible to the naked eye, are accidentally ingested. These eggs typically hide in contaminated soil or on unwashed agricultural produce. Because the eggs are so resilient, they can survive in the environment for a very long time, waiting patiently for a human host to consume them. This stealthy entry method is exactly why poor sanitation infrastructure plays such a massive role in the spread of this disease. Once the eggs are swallowed, the parasite takes a truly bizarre road trip through the human body. You might think it just stays in the stomach, but the reality is much stranger. The larvae hatch in the digestive tract and immediately penetrate the intestinal wall. From there, they hitch a ride in the bloodstream all the way to the lungs. After developing further in the lungs, they are coughed up into the throat and swallowed back down. Only after this incredible journey do they finally settle and mature into adult worms in the small intestine, where they begin stealing your nutrients. Let us visualize this incredible journey with a diagram. Look at the bottom left, where the cycle begins with eggs in contaminated soil. Following the arrows, these eggs are transferred to the hands and swallowed. Once inside the small intestine, the larvae hatch. Notice the path moving upward. They enter the bloodstream and travel to the lungs, shown in the chest area. From the lungs, they move up the trachea, are coughed up, and swallowed a second time. Finally, the large circular inset shows the mature adult worms resting comfortably back in the small intestine. This complex loop is essential for their development. Having seen its complex life cycle, you might wonder why this specific worm gets so much attention in parasitology. The reality is staggering. Ascaris lumbricoides affects an estimated 1 billion people globally. It thrives particularly in tropical areas that lack proper sanitation infrastructure. This is not just a minor nuisance. These worms steal vital nutrients, leading to widespread malnutrition and stunted growth in children. In severe cases, a massive tangle of these worms can physically block a child's intestines, requiring emergency surgery to save their life. By studying this parasite, epidemiologists can predict outbreaks and design effective mass deworming programs. To truly grasp how easily this parasite spreads through a community, we must shift our perspective from biology to mathematics. The environmental contamination caused by a single infected host depends heavily on two factors: the number of adult female worms present in their gut and the daily egg-laying rate of those females. Because these worms are prolific breeders, the numbers get very large very quickly. By using simple mathematical equations, epidemiologists can estimate the invisible threat level lurking in the soil of an affected village. This mathematical approach is what allows public health officials to make informed, life-saving decisions. This brings us to our first mathematical tool, the daily egg production formula. Conceptually, this equation calculates the total environmental contamination generated by one person. The formula is E equals N times R. Let us break down these symbols. E represents the total number of eggs excreted by the host per day. N stands for the total number of mature, adult female worms living inside the host's small intestine. Finally, R represents the reproductive rate, which is the number of eggs produced by a single female worm per day, typically around 200,000. If the number of female worms, N, increases even slightly, the total egg output, E, increases massively. This direct proportionality explains why just a few infected individuals can contaminate an entire village. Now that we have our formula, let us apply it to a real-world clinical scenario. Imagine a young child in a rural clinic is diagnosed with a moderate infection. The physician estimates the child is hosting exactly 12 adult female worms. We are asked to calculate the total number of eggs this child is shedding daily, assuming the standard reproductive rate of 200,000 eggs per female. We chose this linear equation because it perfectly models the biological output. We substitute 12 for N and 200,000 for R. Multiplying these together gives us our final answer: 2.4 million eggs per day. Physically, this staggering number proves that without proper toilets, just one person can heavily contaminate the local soil and endanger the whole community. We have just seen the massive contamination risk from a single patient. Now, let us zoom out and look at the parasite from a broader public health perspective. When a global health team visits a remote village, they do not just look at one child. They conduct widespread stool tests to see how much of the population is affected. To make sense of this data, we need to calculate the disease prevalence rate. This fundamental epidemiological metric tells us exactly what fraction of a community is currently hosting the parasite. Understanding this percentage is the critical first step in designing a successful intervention, as it dictates whether the World Health Organization will recommend mass drug administration for the entire village. To find that critical percentage, we use the prevalence rate formula. Conceptually, this is just finding a fraction of a whole and converting it to a percentage. The formula is P equals I divided by T multiplied by 100. Here, P represents the prevalence percentage of the disease in the community. The variable I represents the number of infected people discovered in the sample. The variable T represents the total population size being studied. We divide the infected count by the total population to get a decimal, and then we multiply by 100 simply to convert that decimal into a standard, readable percentage format. If the number of infected individuals increases, the prevalence rate rises, signaling a more severe outbreak. Let us put this prevalence formula into practice with field data. A global health team visits a village with a total population of 850 residents. After testing, they discover that 170 individuals are actively infected. We need to find the prevalence rate to determine the next steps. We use our formula because it standardizes the raw data into a comparable metric. We substitute 170 for I in the numerator and 850 for T in the denominator. Dividing these gives us 0.20. Multiplying by 100 yields our final answer, 20%. Physically, this means exactly 1/5 of the village is infected. This high rate would immediately trigger a community-wide distribution of antiparasitic medications. We have covered a lot of ground today, so let us review our key takeaways. First, Ascaris lumbricoides is a massive intestinal roundworm that thrives in areas with poor sanitation, causing the disease ascariasis. Second, we learned about its fascinating but destructive life cycle, which involves a bizarre migration through the human lungs before settling in the gut. Third, we saw the staggering math behind its reproduction. A single female can lay hundreds of thousands of eggs daily, making the environmental contamination risk extraordinarily high. Finally, we used mathematical epidemiology to calculate prevalence rates, showing how numbers directly inform real-world medical decisions. You now understand both the biology of this parasite and the public health math required to stop its spread and save lives.